Now we're going to begin our discussion of glycolysis. Unfortunately, I can't get this thing to work in the slideshow, at least presently, so I will set it up for you in D2L. It's a cute little jingle about glycolysis that I think you'll enjoy. So this is the outline for this topic. These are questions you ought to be able to answer by the time we're finished and the time you have studied this material. I'm not going to read through these, but they're for your information, or direction at least. So the complete pathway of glucose breakdown was described in 1940 by Gustav Embden, Otto Meyerhoff, and Jacob Parnas. Glycolysis then is alternatively called the Embden-Meyerhoff-Parnas pathway. It's probably the most completely understood biochemical pathway to date because it has been the most studied. So glycolysis is the first of three linked metabolic pathways that generate ATP, that high energy molecule. It consists of a sequence of 10 enzymatic reactions that convert one molecule of glucose to 2 molecules of pyruvate while generating a net of 2 ATP. So after we've ingested carbohydrates, those carbohydrates are broken down into their saccharide units. Those appear in the blood. Glucose appears in the blood as a result of that breakdown and is transported to cells by specific carriers. Once that glucose enters the cell, it can enter the glycolytic pathway. So remember, I showed you some schemes for enzymatic enzyme setups. These enzymes, again, are only loosely associated, if at all, with each other or other cell structures, so the substrates have to diffuse between those enzymes. So the product, pyruvate, from glycolysis is then converted to acetyl-CoA, which can enter the citric acid cycle, which is the second of these three linked pathways, and is also a source of energy when oxygen is limiting. Interestingly, glycolysis is an anaerobic process. It does not require oxygen, and that means that it was probably developed prior to photosynthesis, which, as we know, produces an abundance of oxygen. There are two phases in glycolysis. We're going to look at these separately. Phase 1 requires an investment of energy and consists of 5 enzyme catalyzed reactions, where each molecule of glucose is broken down into 2 molecules of glyceraldehyde 3-phosphate. I'm sorry, up here. There we go. Glyceraldehyde 3-phosphate. It costs 2 AT molecules to produce the glyceraldehyde 3-phosphate. In phase 2, we have the energy recovery phase. There are 5 additional enzyme catalyzed reactions that convert those 2 molecules of glyceraldehyde 3-phosphate into 2 molecules of pyruvate. The second part of this phase produces 4 ATP, thus we gain a net gain of 2 ATP per glucose molecule. Okay, in this figure, what we're looking at in the middle here are the 10 reactions of glycolysis. It shows the names of the intermediates, the enzymes responsible, where energy is utilized or energy is produced. Structures are shown in this column. We have abbreviations for some of these intermediates. We also have abbreviations for enzymes. And when we draw these together, I will be talking more about those and any cofactors that may be associated with those reactions. But what I want to point out here is that pyruvate, the product of glycolysis, can be utilized in several ways, and that really depends upon the needs of the cell. If we're talking about our bodies, for example, animals and plants. So when O2 is high, pyruvate is normally converted to acetyl-CoA and enters the citric acid cycle, whereby it is oxidized to produce CO2, and electrons are extracted and picked up by electron transport molecules and carried to the electron transport chain. Under anaerobic conditions, so these would be conditions, for example, in muscle, when you're exercising, that pyruvate can be and is converted to lactate. And we'll take a closer look at that as well. So this is lactic acid fermentation in contracting muscle. In yeast, under anaerobic conditions, that pyruvate is converted to ethanol and carbon dioxide and requires a special enzyme in order to do that, one which we do not have, of course. We are not a little yeast. So this is just an overview of phase 1 of glycolysis. There is a regulatory step here, and that is this first step, which requires or involves the phosphorylation of glucose. This requires ATP, so this is one of our energy utilizing steps. This enzyme, which we'll talk about in more detail momentarily, phosphorylates glucose on C6, and we get glucose 6-phosphate. This phosphorylation makes glucose unable to leave the cell. So once that glucose gets into the cell, we want to hold on to it at that point. In the second step, we have an isomerization. So we're going to convert our glucose to a fructose molecule that is still phosphorylated on C6. Then there's an additional phosphorylation, so that requires that second ATP. Now we have a 1,6 fructose bisphosphate, so we have 2 phosphate groups. In the fourth reaction, that fructose molecule is going to be cleaved into 2 3-carbon molecules. Glyceraldehyde 3-phosphate, and dihydroxyacetone phosphate. In the fifth step, which isn't shown here, the DHAP is converted to another GAP3 molecule. So this is the first reaction where we have that phosphorylation of our glucose molecule by the enzyme hexokinase. Hexo meaning 6, kinase means this is an enzyme that transfers phosphate groups. So this hexokinase, in the presence of magnesium is required here, transfers a phosphate from ATP to our glucose molecule in the 6th position. It releases ADP and a hydrogen ion. As you can see, we have a large arrow going in the forward direction, a very small arrow going in the reverse direction. So this forward reaction is more thermodynamically favorable than the reverse. And now we are going to take a break, and we are going to draw all of the structures of these reactions and talk about the enzymes involved, the utilization or the production of energy, and then we'll come back and talk about phase 2.